Hello. Jenna will be joining us via Zoom. She's getting her shot. Dane is here. I've seen him.
Yeah, we just come from analytical, so if we have questions after class to ask.
Okay, good, okay.
I had a question for you.
Yes, ma'am.
So for my thesis, I wanted to include some background information on the two different bacteria I'm using. Is there any particular sources that you would recommend that give like good overviews and information on different bacteria?
On the outer membrane proteins or just the bacteria in general?
Just bacteria in general. Because I feel like the membrane-bound proteins would be a lot easier to narrow down, but there's just like a lot of sources that talk about the bacteria in general. And I just wasn't sure if there was like any that should be used in particular.
There is a site called Micropedia
Okay.
or MicroWiki.
Hey,
Yeah.
Academic Affairs is trying to get a hold of Carolyn. And I don't know what for, but does she have a cell phone?
Yeah. My phone's upstairs. It's in my phone.
When you get a chance, will you let me know what it is?
Yeah.
Okay. She said that she's called her home phone.
Home phone. All we got was bull crap.
So you don't have
People-
that anymore.
People wanting surveys,
Yeah.
people wanting money, people wanting this, people wanting that. Finally, we said, we all have cell phones now. We're getting rid of the
Yep.
bloody
That's what we
landline
did.
phone.
Yep. Okay. So yeah, so if you could get me that, that
Yeah.
would be great.
So now we don't get calls in the middle of dinner.
Yep.
4 of them.
So MicroWiki is a good.
MicroWiki has nice summaries of those different microorganisms.
Perfect. But yeah, I just was like, I looked it up and I was like, there's an overwhelming amount of information. I just didn't know what was good.
Yeah. All right, so we were discussing DNA. We'll get into some biochemical features of DNA. Then we'll look at human genomic DNA and its specific idiosyncrasies. Did you find it, Dane? No. Okay, let's pray. Our dear Lord, we thank you once again for this day. We thank you for the weekend. It looks like the weather's going to be quite nice. Do please continue to watch over us. We pray that Jenna takes her shot quite well. doesn't have any serious side effects, and the protection she gets from it be quite good. We pray now, you guide us through this time. In Jesus' name, amen. So as we said before, when the structure of DNA was solved, it was kind of astounding how a molecule that was so simple could do so much. Furthermore, it became very clear that DNA, whether it came from humans or bacteria or any creature in between, was really the same. So DNA is chemically the same DNA, regardless of its source, which is quite remarkable. So there's only a small number of different things that compose DNA. It has 4 different bases. There's 1 type of sugar and a phosphate, and that's it. And as we said, even though you have this distinct simplicity, you get this remarkable biological diversity. So think of something as disparate as a jellyfish compared to a bullfrog. Think of something as disparate as a rotifer compared to a swan. Think of something as disparate as a human being compared to a paramecium. All those organisms have DNA that is chemically exactly the same. That should astound you. It really should astound you. So what's the difference? The difference is in the sequence of the bases, of course, and that led people to study DNA using denaturation and renaturation studies. Is that big enough for you? So the pioneer of this was a guy named Wilson, Sarich and Wilson at UC Berkeley. And this was their famous study using chimpanzee, great ape, bonobo, orangutan, and human DNA. So they would take strands of DNA, they labeled them, they would denature them, and then they would renature them in the presence of the other species. And then they look for the heteroduplexes. And because only one of the strands was radioactive, and because they used a nuclease that degrades, the so-called S1 nuclease, that degrades single-stranded DNA, only double-stranded DNA would survive the incubation. And because they used very high amounts of the competing DNA, they didn't get any duplexes that were made from the parental DNA. Genetically, that would have been possible. So it was these experiments that led to that number. You constantly hear that human DNA and chimpanzee DNA is 96, 97, 98% similar. It came from the studies of Sarich and Wilson. And then their technique was adapted to study taxonomy in bacteria. In bacteria, Carl Woese used ribosomal RNA genes and ribosomal RNAs by doing renaturing studies with ribosomal RNAs. He was able to make a very sophisticated phylogenetic tree. That tree is largely held up. And then it was applied to other species as well. Now, because sequencing is so fast and so cheap today, this is not done anymore. But for years, it was the mainstay of comparative studies of DNA.
Are humans more similar to any other animals besides humanity?
Extant animals, no. Chimps seem to be by sequence. And the chimp, we have the chimp genome now. We have the human genome. And that we also have the great ape genome. And that's more now. So these studies essentially nicely bore out what these renaturation studies had originally found. And then there were other studies that were done using antibodies to albumin, to hemoglobin, to whatever. Those tended to make trees that were very similar to the trees done with genomic DNA, denaturation and renaturation.
So it's a lot of just people confirming.
Pretty much, yeah, yeah. So this essentially shows you the basic theory behind this set of experiments. So DNA, if you heat it, the bases we know in DNA will tend to breathe, as it's called. They breathe. This breathe is just part breathing, is part of thermal motion of a molecule. All molecules have thermal motion. Proteins have thermal motion, nucleic acids do too. When you heat DNA, you increase the thermal motion. There comes a point where the thermal motion becomes too great for the molecule to be able to stay together. So it will unravel. It unravels usually at the ends first. You get bubbles in the center, it unravels, and then pretty soon the whole molecule is unraveled. At that point in time, we have DNA in random coils. This phenomenon is called DNA melting.
At what temperature?
Depends on the helix. Because length matters and percent GC content matters too. The higher your percent GC content, the higher your melting point. The longer, the higher the melting.
What's like the ballpark range then?
So in these original studies were done, the DNA was sheared. It was sheared by being pipetted through a narrow pipette. So you get fragments, which are very, very big fragments. So the work of Sarge and Wilson shows that the average melting temperature for human DNA was in the 80s. But those are long fragments, very long fragments. And if you renature the DNA, you get sections that don't have homology, you have other sections that do, and so you get these bubbles. And some of the early renaturation studies were done this way. They were done by electron microscopy. So you renature the molecule, you then look for bubbles, you then measure it in the electron microscope, you have a scale. You know how many bases this is? You could reconcile that with a scale, and then you could tell by the percentage of it that was bubbled, that's the amount that's dissimilar, and that's how this was done. That sounds tedious, it's because it is. That's the original way it was done. There's a much easier way to do it.
So when did the easier way come along?
When people discovered the hyperchromatin,
Which was,
I'm about to tell you.
oh,
So the bases of DNA, they do most of the absorption in the UV range. Now, when those bases are in a single strand of DNA, they absorb in the UV range very robustly. Once they're in a double helix, the amount that they absorb because of stacking interactions, the whole helix absorbs, not the individual bases. So now the absorption goes down, and it goes down a bunch. So by detecting the decrease in absorption at A260, you could tell when a helix had melted. Much, much easier.
what's like the time frame for people?
Sarge and Wilson did some of their stuff in the 80s.
Okay.
And they extended that all through into the 90s. Sequencing technology, the high throughput sequencing was being worked on at Beckman Labs and other labs too. what have you. So it took a while, but it was really competition between companies that really drove these. All right, so as you can see here, you have two different species of DNA. And when your drop in A260 is half, that's called the T sub M. That's the melting temperature. That means 50% of your DNA is melted. Then as you keep jacking up the temperature, you get to a point where you're not going to get any more increase in absorption, then the entire helix is melted. Is DNA.
So some genes are
Right.
faster.
Right, exactly. So this one here is at a higher temperature. So that one's either bigger, more GC content, whatever. And so these things essentially, they're the ones that are being studied in this manner. And you can see, based on this type of analysis, you have two types of DNA that have a lot of physical differences. Now, does that say anything about their sequence homology? No. But in the early days, this was the best way you had to study DNA from different organisms. So as I said, the heat stability of DNA depends on the nature of the solvent. So you use one solvent essentially for all DNAs. You use DI water. That's easy. Salt concentration. Higher the salt concentration, but the salt will shield the phosphate more. And it also will tend to, therefore, make sure the molecule stays together, even under higher thermal movement. So your salt concentration is 0. pH is a solvent. High pH, low pH. Now you start to disrupt the bases because the bases are weak. They're weak bases. That disrupts the Watson-Crick structure. So the pH is at ODI water. And then the percentage of the guanosine cytosine base pairs. And then also it's not listed here with the length of the molecule. So the GC base pairs, they're held together by 3 hydrogen bonds. Adenine thymine base pairs are only held together by 2. So the GC base pairs are inherently more heat stable than are the AT base pairs.
So if there's more GC in it, it'll be more.
Yeah. And in fact, this relationship, as you can see, is linear. So as your GC percentage goes up, so does your melting temperature. And a regression line through interpolation and also to some extent extrapolation shows you a nice straight line with a R value very close to 1. So it's pretty much a linear representation, linear relationship. So denaturation will cause a 40% decrease in UV absorption. That's at H260. And that's called the hyperchromic effect. Oh, when it denatures, you get an increase.
Okay.
When it renatures, that's a decrease. What did I say before?
You said decrease.
Decrease.
I was reading-
Decrease when it renatures.
Okay.
I think I was talking about renaturing when the slide said at that time denaturation, and I was talking about renaturation. No. When it denatures, the absorption will go up because the bases in single-stranded DNA, they absorb at H260 more vigorously.
They're more exposed.
Right, they're more exposed. When they're in a double helix, now the absorption goes down, goes down. So that's the so-called hyperchromic effect. And it's clear, kinetically, DNA melting is cooperative. So it's kinetically, it starts slowly, then once it starts, it goes very quickly. So you get bits of the molecule starting to melt, and then once those bits start to join, then all of a sudden, more and more and more of the molecule starts to melt because the other bases are hanging on. It becomes harder and harder and harder for them to hang on. So they start to melt too, and it just kind of all goes at once. So it's cooperative. You get small regions of destabilization. Then after the small regions of destabilization occur, they merge very quickly and you get denaturation. That's why you get curves like this that are so steep. And that temperature at which you have 1/2 of the maximum absorption increase, that's the melting temperature of the T sub M. And if we draw this out, you can see you have a relative absorption at A260. So your baseline is set to 1. And then these are fractions of whatever that reading is here. Because that reading is going to depend on the quantity of your DNA. Then you heat it. You get a line essentially that asymptotes, so it's hyperbolic. So you take the baseline, the asymptote of the increase, that line that falls in half, that's the T sub M. That temperature where this line falls in half, that's the T sub M. When this was all the rage, companies started selling specs that had heated cuvette holders. It would read it, and then it would do this calculation for you and give you a TM. So you could literally do hundreds of DNA samples in a day. Whereas before you were heating, checking, heating, checking, heating, checking. You had to heat next to the spec to make sure the transfer was very fast, and it was a pain. And then they came out with these things. And then right when they came out with that, usually a couple of years later, this was passé. But it was a good run while it existed.
Did you use those at all?
I used them a few times, yeah. I didn't do much of this because I didn't do a lot of taxonomy. So the denatured DNA, if you let it cool, it will renature. If you cool it really rapidly, if you put it on ice, then the DNA will find areas that it can just find homology with very quickly. Those are called snapback structures. They have lots and lots and lots of single-stranded DNA. We used to do that all the time for probes. If you made a radioactive probe, you wanted lots of single-stranded bits. You made it, melted it, and then cooled it really fast, and then used that. If, however, you let it sit and cool very gradually, the DNA will do kind of a hunt for regions of homology, and it's very much a trial and error sort of thing. But once it gets one area that's of good homology, the zipping now, rezipping becomes cooperative, and it can occur very quickly. Just like what you see here. So the overall structure of the molecule, there is room for movement, but if you compare the movement of the double-stranded molecule with the movement you get in the individual nucleotides, movement is pretty restricted in the double helix.
So if you had 2 different samples of DNA and you couldn't sequence them and you wanted to see how homologous they were, could you melt them together and see if they
Sure. Yeah. I used to do a lab with that, but students came back and they complained and they said, why did we do that? Nobody does this anymore. Knowing how to do this is a waste of time. So I said, okay, I don't do that lab anymore. It was an easy lab to do too, but now your bases, as we said, they can move around that glycosidic bond. So you can have two possible orientations, anti or syn. We talked about this already. So here's the syn, here's the anti. And there is movement allowed, but when this is in a hydrogen bonded situation with another base, it's not going to be twirling around like a party trick, essentially. The syn conformation, as you can see, is unstable. There's steric hindrance. So because of the steric hindrance between the bulky side groups and that 5' hydroxyl in double-stranded nucleic acids in the A form and the B form of DNA, all your bases are in the anti-conformation. The exception, of course, is Z DNA. We have alternating purines, pyrimidines, and they alternate in the anti-syn, anti-syn, anti-syn. And when DNA converts from the B version to the Z version, you get the rotation of each purine base around the glycosidic bond from the anti to the syn orientation. The pyrimidines are still in the anti configuration. In zDNA, it's the purines that are in the syn orientation. What about ribose sugar flexibility? Ribose sugars are subject to a bit of flexibility, but not much. So the ribose, you might expect it to be planar, but it very clearly is not. So you have a eclipsing of substituents if it was planar, because those hydroxyl groups would be staring right at each other. They would essentially repel each other greatly. So to relieve the strain, you have puckering of the furanose ring. So it becomes slightly non-planar. That way you get those hydroxyl groups away from each other. So the majority of your DNA molecules, at least that have been crystallized, the ribose is puckered, and it's either C2 or C3 that's puckered, which makes sense. Those are the two molecules that kind of have the most freedom. The others are involved with bonds to either the base or to another phosphate. So C2, C3 are less constrained. Now, if the C2, C3, if they're puckered to the same side of the ring as C5, that's the endo configuration. If they're puckered away from C5, they're in the exo configuration. And in B DNA, all your ribose molecules have a C2 endo pucker. A DNA, all your ribose molecules have a C3 endo pucker. And zDNA is the oddball. So if you have a purine attached, the purine is in the syn conformation, and you have a C3 endo pucker. If you have pyrimidine attached, it's in the anti-configuration, and you have a C2 endo pucker. So the repeating unit of zDNA is a dinucleotide. Sugar phosphate backbone is pretty constrained. You get some movement, but it's pretty small. It has a lot of intrinsic flexibility. That means the molecule can bend. And the sugar phosphate, because of its charge, will play a role in non-specific interactions between things like histones and stuff like that. The sugar phosphate backbone is flexible. How do we know this? When you denature DNA, it forms a random coil. You have to be really flexible to do that. This, I think, should illustrate to you the restrictions that come with being in a double helix. It makes the molecule much more constricted and therefore much more stable. Much more stable. All right, let's look at base pairing. So the base pairings between adenine, thymine, cytosine, guanine, those are called Watson-Crick base pairings. Wasn't really Watson and Crick that discovered them. It was Jerry Donahue, who was in the laboratory, who was a chemist. Apparently Watson never took a chemistry class. Jerry Donahue was an organic chemist. Apparently, when they started with the bases, they had the bases in the wrong form. And the bases could be in a keto form or an enol form. They had them in the enol form. Donahue said that at pH 7, they're going to be in the keto form. Try it now.
But it's helpful to work with people.
Hey, they start willing to listen to people. Thing is, Rosalind Franklin thought the bases were in the inside years before Watson and Crick came to that conclusion. It's only when they got that X-ray crystallographic picture, the mysterious picture 51, that was shown to them by Wilkins, that they said this is a double helix and the bases have to be on the inside. Franklin said the bases have to be on the inside because it makes no sense to put hydrophobic molecules on the outside if you're in an aqueous environment. You have to be on the inside. Just basic chemical logic told her they had to be on the inside. So now, are these bases capable of other hydrogen bonding? Yes, they are. But in a double-stranded molecule, those other base pairings are not stable. But if you make crystals of, say, adenine and thymine, they form very different hydrogen bonds. And this is called Hochstein geometry, after the Dutch chemist who crystallized it. And the Hochstein geometry is inherently more stable than the Watson-Crick geometry. Why then don't bases in DNA use Hochstein geometry? Because of the restraints put on them by being in a double helix. You use what's available to you. So here is, yeah. So here is a Hochstein pair. So if you take thymine, adenine, you get this pairing that's completely different from what you get in a double helix, and this is inherently more stable.
Can you draw it on the board the pairing in a double helix just so it can see side by side?
I can show you. Oh, did I not? Oh, crap. Rats.
So I know it was the first PowerPoint you showed us for this topic. I read it.
Yay!
So,
I take it your allergies have come up.
What'd you say?
Take it your allergies have reared their ugly head.
Yeah. Oh, I think it's like I didn't lost my nose for 3 days. Yeah.
So here is here's the base pairing in DNA between adenine and thymine. And here's so the methyl group's up here. And then there's the Hochstein pairing. See, this is on the other side of this.
Yeah.
See?
Thank you.
So it's very different.
I know it's different, but I want to see how you like.
Yeah. And this is more stable than the Watson Crick pairing. Now, do the Hochstein interactions, are they biologically relevant? They are in tRNAs. And probably in some small RNAs that have various functions. Also maybe in other types of regulatory RNAs. But in DNA, they're not, but they are probably in RNA. Therefore, this does have biological relevance. Now, it's clear that hydrogen bonds are important for the structure of DNA, but they're not the main stabilizing forces of DNA. What do? They hold the bases in place. By holding the bases in place, they can stack. And it's those stacking interactions that seem to be the more powerful forces holding DNA together. So the bases are hydrophobic. Notice the similarity with protein folding and DNA structure. Protein folding, it's the internal association of the hydrophobic bits of the protein that drive protein folding. You fold around that hydrophobic core, and then the rest of the protein finds its structure. Same thing with DNA. You have a hydrophobic core, you assemble around that hydrophobic core, and you twist the molecule to decrease exposure of the hydrophobic core to the aqueous environment. So the bases are hydrophobic. The sugar phosphate backbone is hydrophilic. So if you use ethanol, we'd expect to increase hydrogen bonding and disrupt hydrophobic interactions. So if the hydrogen bonds are the main thing causing DNA to be held together, ethanol, if it increases hydrogen bonding, should raise melting temperature. It doesn't. Instead, it lowers it because ethanol disrupts hydrophobic interactions. That's a demonstration that the stabilizing power of those hydrophobic interactions within the double helix are the main forces keeping the double helix together. So genes are composed of DNA. And we have to clarify this a bit. Genes are composed of DNA, but not all the DNA in human cells encodes a gene. So genes are units that are mutable. That's the old genetic definition of a gene. It's a stretch of DNA that encodes a polypeptide. We'd have to qualify that today. It's a stretch that encodes an RNA. And that RNA may or may not be translated into protein. So there's genes that encode RNAs that are not translated, but they have definite functions.
That's because more recently, how much of the RNA has much more complex.
Yeah. So such a stretch that is transcribed, we call such a stretch a ORF, or an open reading frame. Open reading frame. Now you can have genes that are not interrupted, where you simply transcribe the gene like this, or you can have genes that are essentially put in a row, and you express them like this, you make one RNA with several different ORFs. Or you can have one RNA with one ORF. If it's one RNA and one ORF, that's monocystronic. Monocystronic is the rule in eukaryotes. Or you can have polycystronic RNAs. That's the rule in prokaryotes. So the conversion of DNA into an RNA template, that's called transcription. The use of that RNA to encode a protein, to make a protein. That process is called translation. It's mediated by ribosomes and the messenger RNA. So the flow of genetic information goes from DNA and it's changed to RNA to protein. DNA is used to make RNA, which is used to make protein. The difference is that retroviruses can use RNA to make DNA, which they then use to make RNA and then makes protein. So that's the exception, but it's just that, an exception. This rule where you have DNA going to RNA to protein, that's a rule you can bank on. So that region of the DNA where the RNA is transcribed, where you begin transcription, that's called the promoter. And the promoter will dictate when the gene gets transcribed. So it's a beginning point for the process of transcription, but it also dictates when you transcribe that gene. That's the promoter. In eukaryotes, you have enhancers. These are sequences that bind transcription factors known as enhancer binding proteins. Enhancers do just that. They enhance the transcription of a gene. They increase its tendency to be transcribed. Enhancers in eukaryotes can be moved around and they'll still work, regardless of where you put them. So genes and proteins in prokaryotes are one-to-one. So Charles Janowski showed this years ago by making mutations in the tryptophan operon, then showing that those mutations that had a specific phenotype correlated with the presence of a particular change in the DNA that encoded it and a change in the protein that the gene encoded. So that one-to-one relationship between the gene and the protein it encodes, that's called collinear. Collinearity, that's the rule for prokaryotic genes. Eukaryotic genes are not collinear. The reason for that is that the genes are interrupted by these sequences called introns. Those introns get transcribed, but they're removed before the RNA is translated. The removal is by a process called RNA splicing. The regions that remain that are ligated together and get translated, those are called exons. Exons. Yep. By a machinery called the spliceosome. And this process is called RNA splicing. And RNA splicing allows you to make lots of different proteins from one stretch of DNA. And there are lots of good examples of this. So for example, in the fruit fly, you have a neuronal adhesion protein called DLAR. DLAR. DLAR is a very large intron in the middle of it. In the middle of that intron, there's a gene that's encoded called SCREW. SCREW is a protein that's very important for dorsal ventral signaling in the embryo. So SCREW is actually present in the intron of DLAR. So when you transcribe the DLAR gene, you take that piece out. When you transcribe the SCREW gene, it's just that one little piece in the DLAR intron, and it's not a problem. So RNA splicing allows you lots of diversity of genes. Prokaryotes very often will show overlap of genes. You'll have different reading frames encoded by the same stretch of DNA. Sometimes even the genes will be transcribed from different strands in opposite directions. If the mutation affects the coding region, it might therefore potentially change the amino acid sequence of the protein it encodes. Those are called structural mutations. Structural mutations. So the mutation that causes sickle cell disease in humans, that's a structural mutation. Others will affect the promoter, affect enhancers, affect things that change the way the gene is expressed. The gene is downregulated. The gene's not expressed that much, but the coding region is still normal. Those mutations are called regulatory mutations. Regulatory mutations.
What are some examples?
P-element inserts in the fruit fly, for example, they tend to insert very close to the start site of transcription. So scabrous, the scabrous insert causes scabrous, the gene expression to go way down. So you get a very mild phenotype, but it's a regulatory mutation. You have an insertion, the gene now is this whopping 5 kilobase insert, but it's a regulatory mutation. You also have mutations in the ROSI gene, night-coding mutation. The ROSI gene is actually fine. It's a promoter that's affected. So you have light-colored eyes, not because the ROSI gene doesn't work, but because the ROSI gene is not expressed properly.
Any examples of humans?
Oh, yeah. Yeah. Lots. Frataxin, the frataxin mutations that cause Friedreich ataxia, those are mutations in the intron that affect regulatory sequences. The frataxin protein is fine, it's just downregulated.
It's a lot easier for me to wrap my head around these kind of things.
And Jenna still hasn't joined us yet. All right, this is chapter 24. So the so-called central dogma of molecular biology namely says that DNA can replicate itself. That's DNA replication. We will study that. DNA and then is transcribed into an RNA. That's process of transcription. We'll talk about that a little bit too. Then the RNA, if it's a messenger RNA, is translated into protein. That's process of translation. We're not going to talk about that too much. That's for other classes. So this so-called dogma, essentially, of central molecular biology, the central dogma, as it's called, is probably a bit of a misnomer, but name was given to it by Francis Crick. It's now a well-established principle, so it's worth talking about. That is bacteriophage T2. So here, if you take T2 and you heat it, eventually the DNA inside the capsid undergoes this thermal motion that just explodes the capsid. Yeah, it's a beautiful picture. Exactly. That is a single linear molecule. single linear molecule. And so the T2 phage particle has that capsid head structure, and then it attaches itself using the tail to the bacterial cell. And this shows the DNA that's normally packaged into that headpiece. So this shows you the collinearity of DNA that you get in bacteria. So here is your DNA sequence. Here's your RNA sequence. Notice it's a direct reflection. It's a complement of this strand here. It's anti-parallel to this strand here. So this is a so-called template strand. This is the, for lack of a better term, non-template strand. The RNA is read in triplets, codons. These encode a specific protein. The RNA is read from 5' to 3', from front to back. You then make the protein from amino terminus to carboxy terminus. You make the protein from front to back as well. And each of the sequences in this DNA corresponds to a sequence in the mature RNA, which corresponds to an amino acid in the protein.
So both the RNA and the polypeptide are named like that. Or is the-
Well, yeah, because your translation is coupled in prokaryotes. While you're transcribing the DNA, the RNA tails out and ribosomes jump on it and start translating it. So you literally go from DNA to RNA to protein, just like that.
Okay. Yeah, I guess I never knew that it was like, it could be happening at the same time.
In prokaryotes, it can, because you don't have a nuclear membrane preventing that from happening. So this shows you the genetic content of each of these bacteriophages. VX174 is a single-stranded DNA phage. So this is very small. This was the first thing that was ever completely sequenced, was VX174. It's 5.3 kilobases in size, very small.
So they had to do all the things individually.
Yep. So how long is the viral particle? 25 nanometers, pretty small. So Robert Sinchheiser worked on this. After Sinchheiser became president of UC Santa Cruz, work on VX174 stopped. Then there's phage T7, then phage lambda, and then phage T4. Notice the increase in size of each of these. Notice the length goes up with the increase in size, and the long dimension of the viral particle goes up as well. So here we have an example of an E. coli molecule. That's the whole molecule. That little piece right there, that would represent the size of one of those viruses. Yeah. So the E. coli molecule is 4.2 million bases. 4.2 million bases. And if we bust open an E. coli molecule, here's the DNA essentially packaged in like that. You can see here plasmids in white here. But once again, notice it's organized more or less in domains. The little black specks on there are artifacts of the preparation. So here are some sizes of the genomes of some of these different organisms. Now, once we get to eukaryotes, the genomes tend to get bigger. But I want you to notice something. There's Caenorhabditis elegans. Here's Drosophila melanogaster. Now, in terms of complexity, do these two have any difference in complexity, or are they about the same? Yeah, in terms of number of cell types, they're about the same. Their genomes differ by just a bit. In terms of genes, this one actually encodes a few more genes than this one.
That's so interesting that it includes more genes, but it's small.
Yeah. Now, Drosophila melanogaster, I don't know where they get 18. Melanogaster has 4 chromosomes.
Are
I don't know why they have 18.
any of the other ones wrong?
Okay, that one's correct. That one's correct. That's correct. That's correct. That's correct. Yeah, that's correct. Well, wait a minute. Okay, fine. 23 pairs of chromosomes. So this haploid chromosome number, well, diploid numbers given. Okay, diploid numbers. Okay, that's yeah. Okay, yeah, no problem. Now notice that the rice genome encodes more genes than the human genome, but it's smaller. Yeah. This is called the C-value paradox. The C-value paradox. Bigger genomes don't mean more complicated critters. Once you get to eukaryotes, the dynamics of genomic evolution are not straightforward linear like they are in bacteria. In bacteria, competition, intense competition for speed of division puts bacteria under intense selection to have genomes that are as small as possible. So you have very little space between genes, you don't have introns, that's the case. Once you get to eukaryotes, that does not apply. That does not apply. And so these other sorts of genetic drift now plays a much bigger role in genome evolution. That gives rise to this phenomenon called the C-value paradox, where you get these ferns that have enormous genomes, much bigger than the human genome. And then you'll have pufferfish that really have very small genomes, even though they're much more complicated in terms of number of cell types than maidenhair ferns. So keep in mind with eukaryotes, size of genome does not translate to complexity. Something to keep in mind. Also, genome size is going to vary wildly. So here we have a pair of linked and condensed chromatids, metaphase chromatids from a human cell. And this is at metaphase mitosis. That's the best time to take pictures of chromosomes. If we look essentially at the organization of genes, so here is the hemoglobin, beta globin gene. We can see that we have an intron in gray, an exon in red. Notice that we have these two introns here, and then we have these base pairs here. For the ovalbumin gene, this is from chickens, we have intron. So genes will vary. Organisms will vary as to the number of introns. Some genes, the introns will vary pretty wildly. So this is a breakdown essentially of the sequences in the human genome. So if you look at the amount of DNA in the human genome, that actually encodes protein. It's really small. It's less than 2% of the whole genome.
Seems like such a waste of space.
So 30% of your DNA encodes genes. But of that, a small fraction of it ends up being exons. Most of it is coding radius, introns, and regulatory sequences. Then you have transposons. Virtually half the genome are these transposons, some of which are still active, most of which are dead. Then you have kind of this miscellaneous stuff, some of which still isn't clear what it is, other of which is small sequence repeats, other of which is satellite DNA, 25%, and then 30% of genes.
I mean, I know it's very effective, but it really just seems like such a waste of DNA to have so little of it.
Well, this is common in genomes in eukaryotes. It really is common. You do have genomes that are very compact. Arabidopsis, very compact genome. Pufferfish, wonderfully compact genome. Zebrafish, no. Zebrafish has genomes that are as big as ours.
And they're-
Xenopus. Xenopus is a gargantuan genome. Xenopus genome is maybe 3 times the size of the human genome.
I wonder, like, I'm curious why that. I wonder what factors led to.
Well, with primates, this content is pretty common in primates. So the primate lineage, with it, which if the fossil record is to be believed, soon after the demise of the dinosaurs in the Paleocene, you had branches that led to primates even early back then. So Aethiopithecus is supposed to be the first primate branch. So, yeah. So if we look at the structure, for example, of a yeast chromosome, we notice something. At the ends, you have special sequences called telomeres. And the telomeres can be found based on their sequences, their unique sequences. They cap the ends of the chromosomes. Towards the center, but not always towards the center, you have a sequence called the centromere. The centromere is important for the attachment of the kinetochore during mitosis. In between the telomere and the centromere, you have your genes, your other stuff, dispersed repeats, blah, 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 multiple replication origins, so on and so forth. And this structure seems to hold pretty much throughout eukaryotes. So the work on yeast has pretty much shown this to be the case. Now on Monday, we'll talk about the sequences of telomeres, and then we'll get into supercoiling and what have you. And this will lead us to a discussion of DNA replication, the enzymology of DNA replication, which is very interesting. And yeah, that's where we'll go.
Will there be places?
Yep,
Okay.
just need to write it.
And then I had a question about the lab. So I'm writing